Yeah, you can go ahead.
Okay. So is the slide visible?
Yeah, it's clearly visible from my end. So people please coordinate, and whenever she asking anything, please interact with the tutor. Adesh,
Okay.
yeah.
Okay, so good evening, everyone. I'm Dr. Kirtana Ravichandran, and today, so welcome to the session, and I will give a brief on cell and gene therapy, their regulatory and CMC strategies. So we will go to the talk. So in the title, what does this word CMC short form means? It means chemical manufacturing and control strategies. So it talks, so the talk will be an overview of what cell and gene therapy is and also how is it marketed into the community. So let's go into the overview of cell and gene therapy. So in the rapidly expanding era of biomedical research and increasing genetical disorders, cell and gene therapy has developed just from an hypothesis to a life-saving reality. So what do you mean when we talk about cell and gene therapy? So we are well aware of this picture. So we know that our genes are the blueprint of our physical characteristics. The genes, they contain information in the form of DNA for the production of proteins. A cell's function and behavior depend on which genes are being activated. A slight change in a gene that results in a different protein shape is known as mutation. So most of the mutations are harmless, where they produce a slightly different protein, but they still perform its biological function. So here you can see, I will put the pointer. So here you can see it's a normal gene. Normal genes normally produce normal protein and normal function. But in a case of a mutated gene, it leads to abnormal protein formation or there is no protein at all, which leads to a disease condition. So most of the mutations are mutations are harmless, but some mutations have harmful outcomes. So which means that when a necessary protein is under or overproduced, or if it is produced incorrectly, it can lead to disease or illness, which where the protein cannot fulfill its role in the body. So most of the pharmaceutical products which involve drugs interact with the body's biochemical pathways to achieve its therapeutic effect. But in the case of gene therapy, the aim is to modify the patient's genetic makeup so they can prevent or treat the disease for a long term. So it says the topic says cell and gene therapy. So are they the same? So no. So cell and gene therapy, they are both different. But the similarity involves similar complex manufacturing processes and challenges in patients' access.
So before moving to the previous slide, next slide, just to go back to the previous slide. So is there any questions with anyone? You can interact, Dr. Kitana, so that the people want to ask anything.
Okay. So if anyone has any questions, you can just interrupt in between only and then you can ask.
Yeah. Bhushan, is it clear, Bhushan?
Yes, sir. It's clear.
Yeah, yeah. Please, Bhushan, you support for please interact with her. If anything is there, you can stop and ask. Okay. Yeah,
Yes.
others also allowed. Yeah, anyone.
Yeah, sure.
Just talk because I cannot see who's because of this
Yes, yes,
sound.
Deshman, please. Deshman, please.
Yes, sir. So I just wanted to seek clarification. So when we say the cell and gene therapy, so the basic science is that when a genetic mutation occurs with the cell and gene therapy, the drug goes and then reverse that mutation for a cure to be initiated. Is that right?
Yes. So normally when a mutation occurs, it results in a disease condition. So there are certain diseases which you can see they have symptoms. Okay, so based on these symptoms, then they go for this gene or cell therapy. Depends on what kind of mutation it has been occurred.
Okay, sir. Okay, madam. Thank you.
Okay, so I move on to the next slide. So, what are the genetic disorders? So, genetic disorders are health problems that happen because there are some type of abnormality in a person's genetic material. So, here you can see this picture shows how the DNA is packed into a cell. So, this is a nucleated cell. So, inside the nucleus, they are the chromosomes. The chromosomes are made up of tightly coiled DNA. So, the DNA wraps around the proteins called as histone to form nucleosomes. So, when you zoom in, you can see a double coiled spherical helical structure, which is called the DNA, which is made of units called nucleotides, which has 4 bases, which is the guanine, cytosine, adenine, and thymine. So mostly the mutation occurs because there is a either there is a base pair change or there is a deletion occurs in these nucleotides. So mutation is a change in the DNA sequence of an organism. This change can occur in various ways, such as there may be errors during DNA replication or when there is an exposure to certain mutagens like radiation or some chemicals, or also due to infectious viral agents. So these genetic disorders are grouped mainly into 5 categories. They could be due to any of these factors, like the inherited mutations, where the mutation genetic change is passed from a parent to offspring. Okay. In the case of spontaneous mutation, it may not be normally inherited, but it can occur randomly. So it may be a change in a DNA replication error or due to unknown factors. Then it could be due to chromosomal abnormalities where there is a change in the number or structure of the chromosomes. Then there may be environmental factors where there are external influences like radiations, chemicals, infections can lead to genetic mutation. Then the 5th one is the multifactorial inheritance, which means it could be due to a combination of any of the above 4 or multiple gene mutation. So these are the causes of genetic disorders. So mutations, they have a wide range of effects just from being harmful, or they could be, in most of the cases, it's down regulated, so it is maybe beneficial, but most of the cases it's also harmful to having no noticeable impact on the organism. Certain genetic disorders can significantly increase an individual's risk of developing cancer. So I'm moving to the next slide. If there is any questions, you can just ask. So what is cancer? Cancer is a very hot topic. So hence cancer is fundamentally a genetic disorder, meaning it arises from changes in genes that control cell behavior, but it is not always inherited. Inherited means not always from a family background. So here you can see the picture where there are normal cells. Normal cells, which means that they are normally arranged in a uniform manner. They undergo controlled proliferation and apoptosis. But when there is a genetic mutation, abnormal cells arise, which leads to multiplication and proliferation into abnormal cells, which leads to tumor formation. A mass of these abnormal cells is called tumor. The tumor cells grow in an uncontrolled manner where they start to grow their own blood vessels for oxygen and nutrients, which is called as angiogenesis. Later, when these abnormal cells proliferate in a abnormal manner, they also start to invade into the surrounding tissue, leading to metastasis. While some cancers are caused by inherited gene mutations, most of them are due to genetic changes that occur due to a person's lifetime, which is lifestyle. These changes or mutations can disrupt a normal cell growth and division, leading to uncontrolled cell proliferation and tumor formation. So now there are a lot of treatment process which is available for the control of cancer, which may be due using radiomics or pathomics, where they extract the data from tissues or quantitative data, like they do CT scan, MRI, PET scans, and then they do image acquisition and they lead to treatment and prognosis. Then there is use of nanomedicines where they use nanoparticles, or using extracellular vesicles where they use exosomes and microvesicles, which is extracted from the cell, using natural antioxidants, or targeted therapy or immunotherapy using immune cells which target specifically on the tumor cells or cancerous cells. Then there's gene therapy. Then also there are treatments using thermal ablation where they use magnetic waves or heat waves and destroy these cancer cells. Most of these gene and cell therapy are used for blood cancers, and currently there are 7 FDA approved CAR T cell therapies or cell therapies, which is designed for blood cancers such as acute lymphoblastic leukemia, large B-cell lymphoma, and multiple myeloma. So why cell and gene therapy? So conventional drugs for genetic disorders, primarily they focus on managing symptoms and complications rather than directly correcting the underlying genetic defect. So it's a long-term process when you use conventional drugs, and you have to use it repeatedly because they only target specifically on the biochemical pathways. These therapies often involve small molecules that target specific proteins or pathways affected by the genetic mutation. The examples include enzyme replacement therapy, where they replace the missing or faulty enzymes; substrate reduction therapy, where they lower the levels of toxic substances that can build up during an infection; and chaperone therapy, where these chaperones they help misfolded proteins work normally. In some cases, there are drugs that can modify the gene expression or even facilitate read-through or premature stop codons. But majorly is the manufacturing process and the time taken for the conventional small drug therapy and for gene therapy. So in the conventional small molecule clinical trial, it takes approximately 12 years. But in the case of gene therapy or cell therapy, which is mostly used for the treatment of rare diseases, it takes around 3 to 5 years for the clinical trials to finish. So in the case of small molecule clinical trial, there are phases, which is after the research, it comes the preclinical trial, where they use lab and animal models for to study the safety, efficacy, and toxicological kinetics. Then after the first approval comes the phase 1 and phase 2, where where they mainly focus on healthy volunteers. In the case of phase 1 clinical trials, the drug is tested on healthy volunteers, where they evaluate for the safety, efficacy, and also for the dosage. Then they go for the phase 2, where they use it on a small number of around 300 to 500 patients who has the disease. So this is to assess the efficacy, safety, and the tolerability. Then they go for the phase 3 trial. The phase 3 trial is on a very large sample size, especially like around 3,000 to 5,000 people, where mainly you can see for rare side effects. So mostly in the case of phase 2, where the very small number of people are tested, if there is a rare side effect, it can be missed. But in the case of phase 3, if there is a side effect, these can also be studied in the phase 3, where even long-term follow-up of the patients is done. Once these approvals are done, then they go for the approval of the drug. In the case of gene therapy, there is only 3 phases where you do the preclinical trials using lab and animal studies. Then you pool the phase 1 and phase 2 into one phase, where directly you test it on the patient with the disease. This is because these studies are done only on rare disease conditions, on only certain diseases. So they do directly on the patients with the disease. Then once the dosage and the phase 1 and 2 trial is done, they directly go to phase 3, where large sample size of patients with the disease is tested. So what is the prospect of a cell and gene therapy? Cell and gene therapies are used to treat a variety of disorders, including both genetic and acquired condition. Specifically, they are being used to address cancers, inherited blood disorders like sickle cell disease and thalassemia, neuromuscular disorders like spinal muscular atrophy, and certain inherited eye diseases like the Leber congenital amaurosis. So additionally, there is also ongoing research on other diseases like heart disease, diabetics, and Alzheimer's. And also recently there are a lot of research going on how to treat solid tumors. So here are the key areas where cell and gene therapy is used. So it's used in ophthalmology, where in the case of eye diseases, dermatology in the case of skin diseases, hematology, blood-related diseases, musculoskeletal and neurological disorders, and also in the case of inborn error of metabolism, some metabolic diseases like this OTC deficiency, where there is a deficiency of this enzyme, ornithine transcarbamylase deficiency, which is a metabolic disorder. So now we will go in depth on separately what is gene therapy, what is cell therapy. So first is the gene therapy. The aim of the gene therapy is to use a genetic material such as a DNA to treat the condition at its most fundamental level, that is permanently altering the disease-causing gene. Which means you add a copy of the wild type or the functional gene, or alter the gene so that it produces the normal wild type functional protein. The new genetic material is introduced into the cell using a vector. The vector could be a viral or a non-viral method. Gene therapy, it delivers the genetic material directly into the cell to replace, silence, or introduce gene to correct or prevent a disease. So here is a picture where you see the cell which is treated with either the non-viral methods like using a gene gun, nanoparticles, polymers, liposomes, or using viral methods like microinjection, retrovirus, lentivirus, adeno-associated or adenovirus-based methods. So once the gene of interest is introduced into the cell, they are taken up via the endosomal pathway into the cell, and based on the pH, the acidic pH, these particles are released into the cytosol and taken into the nucleus, where the gene is released and the protein is made, the functional protein is made, leading to normal function of the protein. So for gene therapy, it is also called in vivo therapy, which means inside the cell, where the gene is delivered directly into the body using viral or non-viral vectors. So here are some products which has been successfully used via the gene therapy, like the Luxotuna, Zolgensma, Roctavian, and Hemgenix. Mostly these are used for eye disorders, some for neuromuscular disorders, and in the case of blood cancers like hemophilia A and hemophilia B. So what is cell therapy? Cell
Hello,
therapy.
madam.
Yes, sir.
Yeah, please, the previous slide, yes. I wanted to get clarity on this. So if the gene that is introduced is producing the functional protein, so this functional protein is what is replacing what the other protein is causing, like the disease the other protein is causing. The functional protein is replacing it.
Yes. So,
Okay. And
so either they integrate into the genomic DNA, and so the normal protein is produced, or they are normally synthesized as an episomal DNA. So a DNA which is apart from where they don't integrate into the chromosome, but they are extra chromosomal. So they can produce the normal protein, functional protein in a higher amount.
okay, so in that case, what happens to the abnormal protein that was initially there? Does the functional protein clears the abnormal protein, or um, the functional protein supersedes the abnormal protein in such a way that the abnormal protein is no more functional? How is the mechanism?
Normally, the abnormal protein is in a very less amount. That's why you have the disease, right? So you have less amount of the normal protein. So the
Mm.
protein, so if there is say a mutation where there is a premature stop codon, then the protein itself is not made. So in that case, you don't have the protein at all. Normally the protein is degraded.
Oh, okay.
So
Okay.
you now what happens is you have the functional protein which is in the normal shape. So the function is normal.
Okay. Okay. Now, my second question is with regards to the vector being used. If it is a viral vector, where assuming someone has taken an Ebola vaccine, and then now they are using a viral Ebola vector to transfer that gene, and the person has immunity against the Ebola virus. How is that solved? Especially if the viral vector being used, the person has immunity against the viral vector.
So the vector is only a carrier. It's like a package taking the thing you need into the cell. So normally the virus itself has no immune reaction. So this is normally tested. So mostly they use, lenti and adeno associated virus. The reason is these viruses, they don't replicate inside the body, and they also don't cause any infection. They have less they are less infectious to the humans. So only these kind of viruses are used. The main idea is only how you carry your functional gene into the cell. So it's only a method. That's it. So normally the virus itself doesn't have any role to play inside the body.
Okay, madam. Thank you.
Yeah. Hi, Kitana.
Yeah, hi.
Yeah, this is Vanu Reddy. I just want to know that, see, when you are targeting the cell, means like we have to give your vector.
Mhm.
So means what type of means like administration we are doing? Like it is like cell targeted or like one location targeted, or because your
Yes.
cancer cells might be, for example, blood cancers. Your cancer cells might be floating throughout the body. But how will target to introduce your vector into the particular cell, and then how will control whole body cancer organism, means like things.
Okay, I got it. So normally what they do is if normally it is receptor and ligand mediated. So normally all these methods, they are engineered in such a way that if you want to target a particular cell, they are having the receptor which is target towards the particular cell. So they will bind to the particular ligand in that cell. So, for example, if you need for a particular neuronal cell, then there are receptors on the surface of the virus or on the nanoparticles. And also, in the case of adeno associated virus, there are a lot of serotypes which has been engineered. So, like around 9 to 10. So, these serotypes have a caspid which has a which is engineered that they bind to the specific tissue type of cells.
Okay, okay. You mean to say that whatever the vectors we are making, so those itself having the target mechanism to bind
Yeah.
to that particular cells, and then it will induce the phagocytic mechanism.
Yes, yes. So
Okay, okay.
this is now being developed in such a way that they target specifically on the cell type you need. Before it was like you have also less efficacy because they bind up, to all kind of cell. But now they have
Yes.
engineered caspid, where the virus had these caspids which are very specific to the tissue type you need.
Okay. 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 Thank you.
Yeah, excuse me, madam.
Uh,
In case of for target, in case of a receptor, is a receptor specific mechanism, it's okay. But in case of sickle cell anemia, earlier you mentioned, right? In
yeah.
that case, we need to change entire bone marrow because all RBCs are generated from the bone marrow, right?
Mhm.
So in case of that, how we manage all bone marrow production, it's convert into sickle cell red cells to normal red cells. Is it possible, that kind of treatment?
I think for sickle cell, I don't know whether there is any gene therapy, because this cell and gene therapy is mostly for, yeah, blood related cancers, but on a very particular cell type. So for now, I don't know for whether they have made anything for sickle cell anemia and how they particularly target. I am not sure. Maybe I could read on it and tell you later.
Yeah. Thank you.
On a very particular disease type, I'm not very sure.
Okay. Thank you.
Pratap, there are specific diseases wise also. So those are it is under still research and development stages. Not yet in the popular commercial stages. So it is under clinical trials, and some other trials are ongoing. So it will come upcoming days. We can see that so many things. So how the generic medicines are replacing today with many diseases by biologics and biosimilars. Similarly, in going forward, so these biosimilars and biologics going to replace by this type of cell and gene therapy products. Yeah.
Yes. Thank you.
Yeah. So hope it is clear to everybody so far. Is there anything to further to discuss with Dr. Ketana? Yeah, please interact. Please interact. This is a open session. It should be a interact between yes, Dr. Vanaradi.
No, that done. I just want to say that you can continue.
Yeah. Okay. Fine. Yeah. Fine. Thanks for joining. Yeah. Participate less.
Okay.
Yeah.
No
Go ahead,
issue.
Dr. Ketana.
Okay. So now coming to cell therapy. So what is cell therapy? Cell therapy means you administer living cells into the patient to treat or cure a disease. So these cells are normally derived from a patient itself, which is autologous cell therapy, or from a donor, from a healthy donor, which is allogenic. So majorly there are two types. This is the CAR T cell therapy, which is chimeric antigenic receptor T cell therapy, where the patient's T cells are modified to treat the cancer cells. Other is the stem cell transplant using blood, which is mostly used in blood cancers and genetic disorders. So here in this picture, you can see this is generally how the CAR T cell therapy is made. So if it is autologous, which means you take it from the patient itself. So you collect the T cells from the patient, and then you do a gene editing, where you introduce the modified CAR T cell uh, towards a specific antigen. Okay, normally it's CD19, where it targets the B cell antigen CD19. Then you do expansion. So cell expansion is done. So this is all done out in the laboratory. So you do a T cell expansion, then you do freezing, and then you store. And when the patient is ready for infusion, then you infuse these modified CAR T cells, which is CAR T cells, into the patient. But in the case of allogenic, which means you take it from a healthy donor, so the similarly you take the T cells and then you do gene editing, where you do the CAR T modification on the T cell. Additionally, you also down-regulate the T cell receptor. The idea is because it is from a healthy donor, you have this MHC1 TCR complex formation, which might lead to the killing of the CAR T cell itself. So you down-regulate the TCR, then you do the expansion, then you purify the only the selected CAR T cell with down-regulated TCR, and then you do the freezing, and then you store, and then you give it to the patients. So this is now called the ex vivo method, where the cells are modified outside the body and then reinfused into the patient. So now there are products where they use the CAR T cell for mostly blood cancers like the leukemia, lymphoma, beta thalassemia, and sickle cell, where they use Chimera, Escartica, Zynteglo, and Lentiglobin. And also there are a lot of CAR T cell therapy which is coming in India and worldwide. Some are approved like the NextCAR19 and Quartami, which is mainly for the B cell lymphoma, which has been approved recently in India. And there are some which are undergoing clinical trials. Okay, so like for multiple myeloma and for age-related muscular degeneration. So these are all the products which has been studied in India. So cell and gene therapy is now the upcoming and a new era of personalized medicine. So to just sum up, it is now being called in vivo gene therapy or ex vivo. In vivo is the gene therapy where you package it via a non-viral or viral vectors and inject it into the patient directly. So it's a direct administration of the gene to the patient. Ex vivo is you collect the cells, modify them, and then you reinfuse them into the patient. So here is just a comparison. So the delivery method for the in vivo is using a vector injected into the patient. For the ex vivo, you extract the cell, do a gene edit and reinfuse. Most of the common vectors used in vivo is a adeno-associated virus, adenovirus, lipid and nanoparticles. In the case of ex vivo, you transfect the cell where the gene is introduced into the cell using lentivirus or retrovirus in the laboratory, where it is applicable in the eye, liver, CNS and muscular disorders. In the case of ex vivo, it is for blood cancers, inherited blood disorders. What is the advantage? It is less invasive, which means that you don't need any surgical method. You just do an injection and it is fast healing and it is direct targeting. In the case of ex vivo, it is very much controlled, where you can control it in the laboratory and the cells can be screened. What is the challenge in the case of in vivo is the immune response, where you have mostly the cytokine response in the case of in vivo, and also targeted accuracy, whether you target it particularly to the cell of interest. And in ex vivo, it is a complex logistics, which means you need a lot of equipments and manufacturing challenges, and is also expensive. So this is normally how a drug characterization and regulatory journey is formed. So for a drug to go, it needs a certain developmental stage. So first is the discovery or the research phase, followed by preclinical research. So preclinical research, as I said before, is where you use animal models to test the drug in the initial stage. Then you go for the IND submission, which is the investigational new drug submission, or an FDA review, which is the Food and Drug Administration review. So this is the first regulatory submission which needs to be approved, where they give permission to start the clinical trials, phase 1, phase 2 and phase 3. So as I said, phase 1 is on healthy donors. Now you test for the dose, for the safety, and then you go for the phase 2 and phase 3, where you do the clinical trial on a patient in a small and a large scale, which is then needs to be approved by the new drug application NDA or the biologics license application BLA application, and the FDA review, which is the second regulatory submission which has to be done. Which is then ready. Once the approval is done, then the drug is ready for the commercial manufacturing and approval. leading to post-marketing surveillance. Now we will go to a case study where I will take only one product and we will discuss how it has been done, how it came into the market. So the first is the gene therapy product, which is the Zolgensma. So Zolgensma was used to treat the genetic disorder, spinal muscular dystrophy. So a mutation in the gene SMN1 gene led to a survival motor neuron SMN1 protein deficiency that causes motor neuron loss in the brainstem and spinal cord, leading to weakness and muscular atrophy. So mostly it occurs in the children. So mostly unaffected children, if you see in a normal child, they have sufficient SMN1 protein produced. So there are two isotypes, SMN1 and SMN2 protein. 90% is SMN protein, which is produced for the normal function. And 10% they have a backup gene, which is called the SMN2 protein. In general, the SMN2 protein is only 10%. So in the case of spinal muscular atrophy, the SMN protein itself is missing or is not working at all. So the 10% of the SMN2 protein, the backup gene, is not sufficient. And the amount of the SMN2 protein also determines the severity of the condition. So this leads to the spinal muscular atrophy in children. Excuse. So Zolgensma was designed to deliver a normal copy of the gene encoding the SMN1 protein in patients with spinal muscular atrophy. So this product was launched by Avix, which is a co-company of the Novartis Biologics, where they submitted a license application for the license of this product, which in general is called as onasemnogene abipravovec zeoyoc. So what is the mode of action? So this product uses an adeno-associated viral vector-based gene therapy, which is used for the treatment of pediatric patients who have this disease, who are less than 2 years of age, with spinal muscular atrophy with biallelic mutation in the survival motor neuron SMN1 gene. So the product is administered via intravenous infusion. So the active ingredient is a recombinant self-complementary vector with a double-stranded DNA genome, which is enclosed in a capsid, that is AAV9 capsid protein. So as I said before, so this AAV9 is a serotype of the AAV virus, which is specifically targeted into the CNS, where it can, the main advantage is that they can pass through the blood-brain barrier. The vector DNA contains a transgene encoding the human survival motor neuron SMN protein under the control of cytomegalovirus enhancer and chicken beta actin hybrid promoter. So here you can see how it works. So you have the AAV virus with the DNA, which is a double-stranded DNA genome. Okay, they are self-complementary, and this genome is now packaged into the AAV9 virus, and it is introduced into the cell. So they are taken up by the endosomes, and when there is an acidic pH, these viruses get released out into the cytoplasm and they enter into the nucleus, and this DNA is then now they don't integrate into the chromosome, but they form an episomal or an extra chromosomal DNA, where they lead to faster protein expression, and so the SMN protein is translated and expressed in a normal manner. Is there any questions in this slide?
Yeah, please interact if is there any questions. Yes, Datta.
Is it approved in India?
Jolmens,
product-
I think some people have taken. It's a little bit costly, but mostly in US this is well used. In India, I think it is one or very few patients are under this Jolmensa.
No. That means the question is this product is approved at India to usage or registration?
Yes, yes. There are some patients which I read that there are patients who are taking this product.
Okay. Yeah.
Okay.
So I go to the next slide.
Hello, madam.
Yeah.
Yeah, so with regards to this drugs, the cell and gene therapy, as we said, they are more of personalized medicine. It's based on the genetic makeup of that personal individual that you can choose this particular drug, or is available to, pediatrics as far as you are 2 years.
Mm-hmm.
So my question is, how does the personalized comes in here? Is it based on even though it's a genetic therapeutic medicine, are we still focusing on the fact that it's based on people with specific genes? How do I put the question? People with specific genes that produces that defect, or as far as the person is a pediatric patient and is 2 years and is expressing that particular disease, this drug is okay for them.
Yeah, because it's more difficult. See, most of the genetic diseases, it's you treat them with a conventional drug, then they have to take it for lifelong, and you also don't know whether they are specifically targeting or also it's less effective. So in the case of gene therapy, it's a one-time shot. And as I said, it's less invasive, which means that the treatment method is not very difficult. You just have to give a shot into the patient. Okay, so it's faster healing and more controllable. So it's more easy to treat kids using the gene therapy.
Okay, so let me try and then narrow my question a bit. So assuming this drug is approved in India or Asia, and then there's someone who is from Africa who has a different genetic makeup, will that drug still be applicable to the person, even though it's kind of personalized treatment?
If the disease is common to everyone wherever they are, right? So I can understand. You mean how will their immunity? So this they will follow up. So normally there won't be like lot of people with the disease. There may be one or two people in other countries who wants this drug. Then they will go for the approval accordingly in the hospital. So normally the doctors will recommend the patients, and also the regulatory officials whether the patient can take the drug or not. So normally the doctors will recommend this to the patient.
So I'm asking it because most of the times when we are evaluating dossier submissions, we want to know how expansive the clinical trials has been done, whether it has incorporated people from African descent, so that we know that the drug is good for people from Africa with a different genetic makeup as compared to Asia. So then the question becomes, is this recommendation from the doctor based on the clinical inclusivity of African descent, or it is based on whether the disease is general to everyone, and then we can recommend this drug as far as you are carrying the disease, without considering whether you are from Asia or Africa or Europe or something like that, genetic differences between human beings.
So practically, if you think, I'm not sure whether this is true or not, but I'm telling us normally these kind of genetic disorders happens to one in, say, 1,000 or 10,000 people. So there might be very few patients who come up with symptoms and they need treatment. So normally the doctor will look into how the symptoms are, and then they will treat the patient, right? So normally they will recommend, and they have to do a trial on the patient. If it is a large group of people in the country who needs this kind of, then it will go into a very deep regulatory thing. But I think if there are very few people, then I think the method of this regulation will go different. This is what I think.
Okay, thank you.
Hello.
Hi, ma'am. Okay.
Yeah. This is a CGT based product, is a personalized product. So it is maybe very difficult to perform the phase 3 trial because of the lots of uh, volunteers are required. So how to handle these situations?
Normally, they, if in the case of cell therapy, they directly do only the phase 1, phase 2. So if there are a lot of number of patients, this is what I think, because it's very difficult to get globally like lot of patients to test this the product itself. So especially in the case of cell therapy, they directly take, and 50% it's fine, but 50% it is we cannot expect that you do all the test, to check if the cell therapy is working. If it's
The
very
authority
rare,
can
if it is a very rare disease,
the authority can approve the drug without performing phase 3 study for the market authorization for this type of drug.
So normally, I think the patient itself is the trial here in this case. So they will take this patient's approval that they're also equally performing the testing in these patients. And then they will ask for approval. I think so. I don't know how exactly, like they do it on a very rare disease where very few patients are there. Then they take only these patients as a test trial, and then they try for the approval.
Yeah, I will add up this one. See,
Okay.
that's what there is a one of the condition is there. There is a called risk-based approach because lack of actual patients. So we'll go with this kind of new drugs. We'll go with a risk-based approach. That will be a protocol prepared accordingly. So that will take permissions from the regulatory bodies, and that also required studies and approvals required from regulatory bodies as well, and from the patient point of view also. So see here, if you're not going to treat with that particular patient, the patient will be definitely going to deficiency with that particular type of diseases. So we need the patient also need this treatment. At the same time, regulatory body also required this type of study also. So that this type of called as a risk-based approach in the regulatory. So that will be taking in the permissions and the risk-based approach, and protocol will be prepared accordingly. Then that study will be the treatment will go accordingly.
Yeah, thank you, sir.
Yeah. Yeah, thank you. Thank you. Yeah, go ahead, Kirtana.
Okay. So for John sir, what was the regulatory action? The CBER, they conducted a preclinical. CBER is Central for Biologic Evaluation and Research. They conducted a pre-license inspection at the manufacturing facility, and FDA Form of 483 was issued, and all the inspection was resolved, and they were classified as voluntary action indicated. So a team of biologics, they performed this at the AVIX group testing site, and the form 483, which is a notice for inspectional observations, was issued, and the inspection was classified as no action indicated. Then they went into this Office of Regulatory Affairs and CDER, which is the Center for Drug Evaluation and Research, which was performed for a survival inspection. And all these issues was resolved, and the inspection was qualified as no action indicated or voluntary action indicated. Additionally, non-clinical pharmacological studies was also proved, where they did in vivo pharmacological studies in SMN delta 7, a murine mice model for the SMA condition. So these mice models were conducted using non-clinical vector also, where they demonstrated that there was an improvement in the motor function, neuromuscular transmission, the body weight, and cardiac function. So once the phase 1 and phase 3 trials were done, and after the ongoing trials, as well as for the patients treated through expanded access program, they contribute to the safety database. Based on these phase 3 and phase 1 trials and the reviewer's team's recommendation, the final approval of Jolmensa for the treatment of pediatric patients less than 2 years of age was approved. So any questions on this first case study, or then I will go to the second one. Okay, so I will go to the second case study, which is a cell therapy product, which was approved in India, which is the Cortami. So Cortami is varnim-cabdagene autoleucel or Varcel, which was commercialized in India. So Cortami is India's first affordable CAR T cell therapy for adult B-cell non-Hodgkin lymphoma, which is a personalized T-cell immunotherapy, which involves extracting the T cells from a patient, genetically modified them into target the cancer cell, and reinfuse them into the patient. So what is non-Hodgkin? So lymphoma is of two types, non-Hodgkin and Hodgkin. Lymphoma itself means tumor of the lymphocytes. So where you see the bulging of the lymph nodes. In Hodgkin lymphoma, you see this rare enlarged dinucleated Reed-Sternberg lymphocytes, where they are high in number and were abnormal in function. In the case of non-Hodgkin's, these Reed-Sternberg cells are absent, but there is a high proliferation of B cell and T cells, which leads to a tumor formation in the lymph nodes. So what they do in this CAR T cell therapy is that they isolate the T cell from the patient, and they genetically modify by introducing a chimeric antigen receptor into the which is specific for the CD19, which is the antigen present on the tumorous B cells. So these CAR T cells, they specifically bind to the CD19 ligands on the tumor cells. So these CAR T cells are nothing but, this CAR is chimeric antigen is nothing but it has a flexible single chain variable fragment, which is specifically antigen which is called ectodomain, which is specific to the antigen binding CD19, which is then linked to a spacer for flexibility, which is then linked to a transmembrane domain and endodomain, which is present inside the cell, which contains a co-stimulatory molecule and CD3 zeta for signaling into the T cells. So this then releases cytokines and other toxic substances from the T cells to kill the tumor cells. So how is it manufactured? First is that you take the T cell extract using hepapheresis, where the T cells are, specifically the T cells are isolated using this machine, where there is a high speed centrifugation is done, where the blood components are collected into these separate bags. So you have, so only you need T cells, only the T cells are selected, and the remaining is reinfused into the patient. So the collected T cells is then removed, proliferated, and then they are genetically modified to express these CAR antigen on their surface. So this is designed specifically to a protein called CD19, which is found on the surface of B-cell lymphoma. Then you do the CAR T cell expansion. So these genetically modified CAR T cells are multiplied in the lab to create millions of cancer-fighting cells. Then the formulation is done in a vial and they are cryopreserved. And once the patient is ready for reinfusion, it is reinfused in the patient. Before reinfusion, so chemotherapy is done to the patient to remove all the cancerous cells in the body so that also there is no immune reaction against the CAR T cells. After the chemotherapy, then you reinfuse these CAR T cells into the patient's bloodstream where they specifically destroy the cancer cells which is expressing the CD19 antigen.
So, madam.
Yeah.
So this collection of blood can be done collected from both healthy and people with the disease, and then later on modified and then re-injected.
Mm-hmm.
Okay, I just wanted to be clear.
No, it depends. So if it can be taken also from the patient itself, or if the patient has very less, in some cases, if the disease is in a very advanced stage and you don't have enough T cells, then there is also possibility that you can take it from a healthy donor.
Okay, so before it's being taken from a patient, then the patient must have a lot of T cells before that process can be done.
Yeah.
Okay, all right, thank you.
Yeah.
I have a doubt.
Right.
Yeah.
So basically, like companies like Immunil, ImmunoAct in India, so do they have to get associated with hospitals?
Yeah, normally they are associated. So this quarterly was done in Immunil, right? So they are associated with Narayana Hrudayalaya. So this is the hospital which they are, where they take the samples and then they do also the trials.
Okay. So is there
Yeah.
like hospital has to follow certain regulatory paths or any like approvals? Like does hospital also get enacted with the company and has to follow regulatory pathway, or that is whole a different section for a hospital?
No, they also have to. So when you submit, you have to tell also whether you have the required laboratory facilities, whether you have also doctors who will look into this, trial phases where they will check regularly on the patient. So all these has to be submitted. So this has to be mentioned in your regulatory submission. So the protocol has to be followed by the lab and also by the hospital or the institute where this research is going on.
Okay. Thank you.
Hello, Kirtana.
Hello.
So what about the product Quatami? What it contains? It contains the CAR T cells from the healthy donors, because this looks like the original personalized medicine where the patient's T cells are taken out and they are being maybe modified. So what the product contains actually?
It contains the T cells, the CD4, CD8, the T cell itself, but they're modified containing the CAR antigen on the surface. So you have
So they are from the
certain,
healthy donors.
most of the cases it is from the patient itself. They mostly try because then there is no immune reaction. This host cell reaction is not there. So mostly they are from the patient itself. So in the case here, you take the cells from a patient who has this B cell lymphoma. So you take
So the
the
product, okay, the product will be made only after a patient's encounter then.
Yeah,
Once
I will
the
add up.
patient
Yeah,
comes.
I will add up to a little bit to this. See, which is the cells identified as non-function in the patient body. Those cells will be taken out, and the genetic systems will be modified in the outside. So the cells which is genetically modified, and repaired. In general, we call as a repair in the terminology of normally generic word. So the repaired cells will be cultured outside. Then after making in the culture, adherent culture, so then we'll go for the how like we are making a biosimilar biologics. Similarly, these type of cells is made in the outside and the culture based methods. After that, we'll go for purification and formulation. So that will be injected in the patient body. So that will be work as like earlier in the function, which is the function is lost in the cells. That will be come back to the treatment of the particular diseases.
Yeah,
So
I understood that. Yeah.
So normally what you do is you take from the patient, they tell you like how many amount of T cells you need. For example, 1 into 10 power 6, 1 million cells or 2 million cells. Then you try to collect 2 million cells from the patient, which takes around 4 hours normally for this apoptosis to occur. So then you collect these T cells. If not one, first day you don't have enough cells, then you go for the second day where you still try to take. In the case of stem cells, what they do is they give some drugs, so there is more stem cells produced. So in the case of CAR T cells, you need only lymphocytes, only T cells. So in that case, you cannot do anything. So how many other T cells is available from the patient? They try to collect as many as possible. Okay, then they collect these T cells. Once, if you have enough number of T cells, it's fine. Then they try to go for the lab. They take it to the lab and they try to expand the T cells using the CD3, CD20. There are specific antibodies and interleukins, these cytokines, which will help in proliferation of these T cells. Then they try to take as much as possible, and then once you have enough number of T cells, then they try to do this genetic modification outside. So they use retrovirus or lentivirus based methods to introduce the CAR antigen receptor on the surface of these T cells. So that is what you see here, right? Then these are selected and then grown in the lab, in the culture bottles, and then they are reinfused.
Okay. Thank you.
Okay.
Hello, madam.
Yeah.
Yes, I just need clarification on from where the culture is done and then the chemotherapy is done. So after the chemotherapy, does the drug go through regulatory process of maybe approval before it is being reintroduced into the patient's bloodstream, or immediately after the therapy? I mean, there's no regulatory involvement after the chemotherapy, and then it is straightly introduced into the bloodstream of the patient. I want to know where the regulatory staffs come in after the chemotherapy.
No, normally, you see, clinical trials is done on the patient itself. So they have to take up to check whether the drug is working or not, right? If not, then how do you check? So they have to undergo chemotherapy, they have to reinfuse the product into the patient, and then the patient is studied over a certain period of. So normally you have side effects, and all these are checked for around 3 months, and then you check whether the CAR T cells are fighting against the cancer cells. So then all these data is collected. If there are a large number of people, then you collect all the data and ask for the approval from the thing. Otherwise, as sir said, in the risk-based method, 1 or 2 patients, then whatever results you have, you just have to submit it to the FDA for approval.
Okay, so madam, if I get you clear, so after the chemotherapy, then the phase 1, phase 3 clinical trial starts, and then based on the report, the drug can now be approved for it to be injected into the bloodstream of patients who are actually suffering from the disease.
They have to, because the patient is already suffering, so they have to do the test anyway. That is what, like sir said, you have to just take a risk and they have to inject the product into the patient.
Okay, thank you.
Hi, I have two questions. Can you go to previous slides? See, if you see that what are the flow of the preparation of the T cells with CAR T, then we are collecting, if in case if you are collecting the blood from the patient, and then how you are going to segregate that T cells are in good condition, or they are in the infected state, means like there is the tumors, like. And the second thing, is it mandatory to do chemotherapy before giving these CAR T cells to the patient?
So normally what happens in the for the first question, you just take all the T cells, whatever is available, whether they are healthy, because you need as much as possible. The rate of the number of T cells needed at the final injection is very important. So based on the first, how much of a T cells you're taking, only then you can do the proliferation in the lab, right? So you need as much as possible. So whatever T cells is available, you have a mixed population, the good cells, bad cells, everything. Normally, all these are automated. So all the cells are collected as much as possible. And then in GMP is followed, where all the T cells are proliferated and the genetic modification is done in whatever T cell is available. So
Okay.
they don't very
Yeah, but
specifically say.
in case if the T cell whatever we have taken, in that if already some cancerous cells are there, in case,
Mhm.
so will it impact means like your whatever the means like CAR T preparation, and then when it is injecting to the patient, how will be the impact?
Uh,
Because we are not defining that these are means completely good cells or bad cells. So
no,
how we are going to, yeah.
they don't separate, good cells, bad cells. But in this case, in B cell lymphoma, it's the B cells which is infected, not the T cells, right? So that
No,
normally you
no,
have
that's
T cells,
okay.
which is
I'm not arguing about the actual disease, but when whatever the T cells you are taking, so these T cells, whether we are going to use for the therapy, and whether they are in the good condition, or they is I mean the T cells itself having some means like already mutations or something. So how will going will define, or whether we'll characterize it, or just we'll take the pool of population, T cell population, and then they will modify and then we'll inject. That is
Exactly,
what I
we'll
want.
follow isolation.
they take a healthy donors T cell,
Mhm.
they then it depends on how advanced the stage is. So if it is in a
Okay.
very advanced stage, then the treatment goes through a allogenic, where you take it from a
Okay.
healthy donor.
Okay. Okay. Yeah. And the second one, that chemotherapy, whether is it really required?
Yes, yes. Normally they do the chemotherapy so that they try to suppress, the immune response against the CAR T cells. Okay, especially in the case of allogenic, they need it, and also that they also try as much as possible to decrease the already infected B cells. So in this case, the infected B cells are all removed because there is a chance of reoccurrence, right? So if there is a reoccurrence, then these CAR T cells will be a savior in the next
Mhm.
production of these lymphoma cells. So for
Okay, you
that
want to
case,
say
chemotherapy
that
is
like
compulsory.
okay, whatever the CAR T therapy you are giving, it's not like that direct means like
It's not the
one
only
time, yeah,
method of,
only therapy,
no,
yeah,
no, no,
to
no.
remove all the cancers.
They
Okay,
have
okay.
additional medicines which are given along with the CAR T cell.
Mm-hmm.
Yeah, but major will be the CAR T cell which will act, and others will be to reduce the off target and also the side effects.
Okay, understood, understood. Thank you.
Hello,
So,
madam.
yeah.
Yeah. So just a quick one. The CAR T cells given to the final patient, assuming we are supposed to start the whole cycle again, can that final patient T cells be taken again and then run through the cycle?
You mean they be given again? You are asking?
No, you see the final person in the previous slide, the final person who took the CAR T cells that has gone through the chemotherapy. That is the final stage. The new CAR T cells introduced into the bloodstream. to the
Yeah.
final hand, can that person's blood, which contains T cells, be taken and then go through the cycle again? Or it always has to be a new person? I mean, the blood of someone who has received a chemotherapy treatment, can the blood of that person be taken and then subjected to T cells separated, T cells modified, and then proliferated, and then chemotherapy applied again and then injected to a new person?
I'm sorry, I don't get the question. So normally you do the chemotherapy. So after the chemotherapy, the new CAR T cells are then introduced. So what do you mean, a new patient?
So my question is, the final patient at the last stage who receives the CAR T cells into their bloodstream.
Mm-hmm.
Yes. Assuming we are supposed to take, the healthcare providers are supposed to take the blood and obtain the T cells again. Can the T cells be taken from that person who received the CAR T cells, the chemotherapy, how do we call it? The T cells that have been subjected to chemotherapy.
From where you take the T cells? No, the healthy donor don't need these. Only the patient will be given, right?
No, not the patient giving. I mean, if the last person in the diagram, that is new CAR T cells introduced into the blood, yes, that patient. Okay, so can the blood of that patient be taken and subjected to the cycle again?
You mean if there is a reinfection, whether in some cases, if the treatment is, it's not working, then they again go for the next cycle. You need to do it.
With a different person or with that same person?
With the same patient. It depends. Again, they check how much T cells are there, whether you have enough. Otherwise, they take it from a healthy donor. So it depends on the result, like whether if the CAR T cell has not worked, then you're asking whether the patient sample will be taken for a second round of CAR T therapy, right?
Yes.
So it depends whether the patient has enough T cells again to give. If not, then they go for the donor therapy.
Okay.
So it depends on the patient's condition. So Quatermain was approved by the Central Drug Standard Control Organization. They were evaluated rigorously, and the Apex regulatory body of pharmaceuticals and medical devices approved for the successful completion using this image trial, which is a part of a clinical trial which specifically does counseling on these social and patients trial methods, which demonstrates the safety and efficacy of Quatermain in treating the adult patients with B-cell non-Hodgkin lymphoma. So the image trial was conducted across multiple hospitals like Narayana Hospital, Narayana Hrudayalaya, the Apollo Cancer Hospital, and PGIMR, which showed impressive results. So an overall response was around 83.83% at the day 90, which sets a new benchmark for CAR T-cell therapy in India. So there was no patient who required an ICU admission, or they did not develop any neurotoxicity, which indicates that there is a favorable safety profile similar to other approved CAR T-cell therapy worldwide. So the CDSCO's approval for Quatermain was based on this positive clinical trial, which marks a significant step forward in bringing this revolutionary treatment in patients in India who are in need. So just a quick comparison of what is the manufacturing, part between a CAR T, which is between a cell therapy and gene therapy product, and a biological or a small molecule therapeutic protein. So in the case of CAR T cell, you go for the apheresis collection, which means you collect the specific cell type you need using this apheresis device, which is automated, and you collect it. Then you do this washing, fractionation, T cell selection, and activation. So you activate the T cells using specific antibodies and cytokines. Then you do this gene transfer where you use viral vectors or transposons or mRNA ectoporation to introduce the gene into the cell. So these are the upstream process. Once the genetically modified T cells are ready, then you'd go for the CAR T cell expansion, CAR T cell formulation, cryopreservation, and infusion into the patient, which is the downstream. But in the case of the small product or therapeutic protein, biological protein product, the upstream involves the isolation of the gene of interest, and then you do an introduction of the gene into a expression vector, and then you transform them into a host cell, where then you use you culture them into a master cell bank or a working cell bank. Then you do a selection of the required sequence and propagation of cell, and then you go for the downstream where you isolate, purify, you extract, then you enrich or isolate the protein of interest, and then you do the formulation, packaging, quality control, sterilization, viral decontamination, and pyrogel removal for the final product to be released. So
yes, ma'am, like on the back side. Yeah. So how much time duration we are talking about for this CAR-T development? And is it in a completely hospital setting or the GMP is involved, like industry?
So normally for the CAR T cell, now people are doing it within 24 hours. So everything is automated. You put it all in a machine, and then there are automated, semi-automated, so everything is done in a machine. Now people are trying to do it within 24
GMP,
hours.
the straightforward GMP and hospital coordination, it is happening.
Yeah.
Because decision is coming from hospitals and making and purification and formatting by organizations like who are having a biological backgrounds. So those are typed with hospitals. So based on the requirement, so they will make it and give it. So it is required to perform under grady areas where the GMP controls there is happening.
Okay.
Yeah.
Thank you.
So for the manufacturing of a gene therapy product, so you undergo this standard procedure. So you take these HEK cells, you culture them, you do transfection of the viral products to form a recombinant vector, in this case the adeno associated virus. Then you go for the downstream where you do the lysis, remove the virus, collect the virus, and then you do all the purification process, and then it is formulated and then vialed into a product. So in the case of cell therapy, in this diagram they show for stem cells, where in this stem cell therapy you need to engineer these stem cells. Then you do a seed train where you stack them into a large amount of cells. Then you do a large scale expansion, cryopreservation, packaging, and harvest. But in case of CAR T cell, then you don't need to go to seed train. You just only do an expansion, and then you directly go into selection and cryopreservation and packaging and harvesting and reinfusion into the patient. So in general, process involves the upstream and downstream processing. Upstream processing means you do a cell growth, product generation, cell line development, culture them into bioreactors, transfection, transduction to get the genetically modified product. Then you maximize the product and use typical equipments like bioreactors, incubators, culture systems, and then you use the crude biological material. And for the downstream, mostly it is a purification, how you formulate them into a final product. Then you do filtration, formulation, you check for the purity, safety, stability of the drug, and you use equipments like filtration product, chromatography column, sterile filing, and the product state is a purified, formulated therapeutic product. So what are the regulatory challenges? In general, you need proper institutional and organization planning for the clinical trial. So you need proper laboratory facility and collaboration with an hospital or institute where they follow good manufacturing processes and GMP facilities, and they have to get approval from the institutional biosafety committee and as to have a proper study protocol which has to be approved by this biosafety committee. Then there is the review committee on genetic manipulation, because you genetically manipulate the cells to treat the patient, right? So you need the approval from this review committee for genetic manipulation, where you have to give information on the import, export, and exchange of all the recombinant DNA products in India. Then, if you use stem cells, then an approval should be obtained from the Institutional Committee of Stem Cell Research. If you do animal studies, then approval from the animal ethic community should be provided for preclinical testing of the gene therapy product, which is followed by institutional human ethical community, because you have to do the phase 2 and the phase 3 clinical trial on human patients. So you need approval from the human ethical committee, followed by gene therapy advisory and evaluation committee for the gene therapy application, and they have to provide recommendations on how this gene therapy product has to be used. Then followed by finally by the CDSCO, which is the Central Drug Standard Control Organization, which will approve for the product. In case you need foreign collaboration of the product, then you need approval with the Health Ministry Screening Committee. And for all the GTP trials, Clinical Trial Registry India registration is mandatory. This is the flow chart of how you get the final product out. So all the control requirement has to be informed, like what kind of recombinant viral vectors are used, what kind of, in case you use non-viral vectors, what kind of non-viral vectors are used, what kind of methods are used for these introduction, and then whether you use microbial vectors, whether they are oncolytic viruses, what are the gene modifying techniques, whether you use CRISPR based methods or short hairpin RNA method of gene transfer, and what are the other genetic materials that is being used, all has to be enclosed in this regulatory procedure. So in that case, all the control focus will be on the raw materials used, who's the donor, what is the cell source, what kind of vectors are used, what kind of controls have been done, like checking for the cell viability, what is the transduction efficiency, what is the culture monitoring, what kind of environment is used, and then the equipments, all the equipment qualification, calibration, and validation has to be provided. And the product testing for identity, the sterility, the endotoxin, whether they're endotoxin free, what is the safety procedures that has been followed, what is the storage validation, what is the shelf life of the product, all these has to be documented. And the batch records, how many batches have been used, what is the deviation, what is the traceability, and all the regulatory compliance has to be done to get the final product approval. So in the case of stem cell therapy, they also need details on the master cell bank and the working cell bank. So master cell bank is where you provide the information on what kind of cells are taken, the source, what kind of methods are used for the production, the stability, the characterization of the stocks that is being used, like on the cell number, what is the density of the cells that is used, and you have to provide the unique identification number, which are all being enclosed in the CMC requirement of the gene therapy product. Also, along with it, the master cell bank has to be tested and documented that there is the absence of pathogens, viruses, like this Epstein-Barr virus, hepatitis C viruses, and all these are absent in these master cell banks. Then also working cell banks testing has to be done. So multiple vials of the master cell bank are taken and used as a working cell bank, which also has to be tested for the in vitro adventitious viral agent testing. So they have to test whether these viral viruses are not toxic to the toxic in vitro toxicity of the virus, that they don't replicate, so they're replicant competent. Then there is no bacterial or fungal contamination, mycoplasma testing, and also for the identity, which means that the product is original, which is normally tested using southern blot and flow cytometry. All these regulations has to be in compliance with the ICH Q5D guidelines, which is a guideline for biotechnological products. Then for the final gene therapy product, you need all these information. So all the product name, the manufacturer of the product, what is its dosage form, what is the route of administration, what kind of vectors is used, what is the target disease that has been used for this product, what is the active ingredient, what kind of materials are used, what is the in-process control, the purity, the potency, the titer, what kind of syringes are used, what is the sterility, the bioproduction, whether there is any toxicological results, all the trial summaries, labels, the patient, the name, then the instructions which has been provided to the patient, and what is the risk management, which means how long are you going to monitor. And then you need all these certificates, guideline adherence, GMP certificate, and then for the viral safety, that they are not replicant, they don't replicate, they are replicant competent, and for the genome integrity. All these certification has to be provided before the final gene therapy product comes into the market. Globally, you can see that various other countries have their own guidelines, which has to be which has to be produced for the product to be done. So like in India, you need to tell what kind of so these are normally the DPT and ICMR are the regulatory authorities, but in the case of USA, it's the FDA. And in Europe, the EMA, and in China is the CDE. These are the regulatory committees which approves the product. And here you can see in summary, like what are the different guidelines or requirements needed in different countries for the approval of a product. And also there are different acts which are used in different countries for the approval of this cell and gene therapy product. So for the translation of a gene therapy product to a new drug, along with the preclinical requirements, you also need the clinical requirements, which means that you have to tell about the disease information, the selection of the patient where you are going to do the trial, the study design. So how you're going to do, so starting from how you're going to take up the cells, or you're going to introduce, how you're going to follow up, all the study design has to be mentioned. So what is this dose selection, the administration criteria. So in case a person is withdrawing from the study, then what kind of criteria is being followed for that, and then whether what is the trial endpoint. So how long is the trial going to last, and follow up is done. So all these has to be mentioned. Along with it, the safety endpoint and also the efficacy. So the patient history, the patient experience, and the follow-up time period has to be mentioned. So in summary, cell and gene therapy is a very promising field of biomedical research for and improving the safety and efficacy of the gene therapy is what is needed for the hour. So cell and gene therapy products differ from conventional biologics in manufacturing process due to their specific characteristics such as the need to preserve the quality of the entire cell as compared to conventional biologics where they focus on the quality of the therapeutic protein product. So in United States and Europes, they are the major ones who are very focused in this field, where the clinical trials have been carried out for more than 2 decades. And now India is stepping up into this field with a much more awaiting results. So many guidance documents are available, like this PICS Annex 2 for advanced therapy medical products to guide the manufacturers and the researchers as new scientific knowledge continues to emerge and evolve. So ethical issues observed in the past related with the use of the gene therapeutic technology led to the development of stringent guidelines because there is a misuse of these products, especially in the case of germline therapies, where they take these cells, stem cells from, pregnant women, so they can use it for some other treatment. So all these guidelines and regulations are made very stringent, so there is no misuse and premature commercialization is done. So with that, I end the session. And in the upcoming, if more people are interested to know, then these are the topics which will be covered in more detail. So whatever is done is just a very short, brief study which has been mentioned to you. But now, if more people are interested, then we can go into more detailed topics, starting from the basics of what cell and gene therapy means, what kind of vectors are used, more on the lentiviral or adenovirus or viral vectors that has been used, the gene editing processes, like now the upcoming is this CRISPR-Cas9 based gene editing. So all these can be studied in more detail, along with the regulatory guidelines, like how you write a dossier, what kind of guidelines is needed, how we submit it to the officials for the approval. So all these can be done. So this is just an outline, like what will be in the coming sessions. If more people are interested, then we can discuss in more detail. With that, I'd like to thank everyone for attending the session. And if there is more questions or discussions, you're more welcome. Thank you.
Thank you. Thank you very much, Doc.
Before going to the questions and answers, so I will add up 5 more minutes session. Kirtana, can you stop your screen?
Sure, sir.
Yeah, so how is the session so far? Yeah, so I'll kindly requesting you, whoever want this session recording, so please drop your mail ID. If already people given, just even though in the inbox you share it, so that we'll post the message. Yeah, Kirtana, just stop your screen. Yeah, thank you. So I want to add up a few more things regarding this session, what are we discussing today, so that it will be aware why we are giving the sessions. So we know that the biologics and the biosimilars vaccines, we already giving a sessions for the people. So similar way, we are going to start one more cell and gene therapy sessions on the online. Are you able to see my screen?
Yes,
please
sir.
respond. Yeah, fine. So whatever cell and gene therapy products also will go with similar pathways. So how we are seeing discovery phase, then preclinical trials if you conduct, then applicable clinical trials, then we'll go for the marketing authorization filing, then we'll launch our product in the market. So the product will be available in the particular organization with the type with any hospitals. So the way of approach for regulatory submissions remains same. So that will be as per ICH guidelines, and as well as especially for the cell and gene therapy country specific guidelines. So with respect to module 3, so again we are going to take up complete cell and gene therapy process. So minimum 2 to 3 products case studies. So how we are going to collect the cells I will go for the genetical modifications. Then I will characterize the cells. I will go for the culturing and purification methodologies, formulations, and how we are going to place in the module 3 those here. So again, the respect to non-clinical and clinical will be given overview, and how we can make the dossier preparation. So especially the dossier preparation with respect to model 3. So at present, so what are the M4Q guideline? It is under draft stage. That is going to come up with the relevant related to the cell and gene therapy as well. So what are the current CTD M4Q guideline? Is not nowhere anything mentioned regarding cell and gene therapy. So to include the cell and gene therapy products, there is a revision is going to happen. So upcoming in the next year, maybe 2026. So that will be coming up with cell and gene therapy introduction also in the M4Q guidelines. So with coming to the regulatory filing and all in the FDA or EMA, so how it is going to happen also, case study will be there. So with respect to FDA, so how these cell and gene therapy products so going to happen in the registration process. So we are going to take up completely as you like biosimilars, biologics, and vaccines. So if you see here, so this is the complete overview of the picture how the products are being submitted in the regulatory filing. So this is how we are discussing about biotechnology products and any recombinant products or any this related to the biosimilars and generic medicines. Similarly, we're having a CBR, that means Center for Biologics and Evolution and Research. So there is a Office of Tissue and Advanced Therapies. So this is the main branch. So there we are going for the filing activities. There will submit the cell and gene therapy products. So these cell and gene therapy products we want to file. So what is the requirement? The requirement, so whatever she explained details, so regarding cell therapies and regarding gene therapies, so we have a scheduled sessions. The people wish to learn this type of online sessions with complete case studies on the cell therapy products, gene therapy products. We are going to conduct online sessions. So how we attended today, similar way. So they are going to conduct around 20-hour sessions. So in these 20-hour sessions, so we will explain completely the products we are taking examples and how we are going to generate the CMC data, and I will go for the filing activities. So we'll take up the case studies how to prepare this type of products in the manufacturing, and start from R&D, then manufacturing, and how to release in the market, and how to prepare the dossiers, how we are going for the filing activities. So those and all will be covered here, and followed by, especially we'll take up EMA and respect to FDA and any other regulatory markets like MHRA, any Health Canada. So as well as in India also will take up a case studies. So here, especially cell collection and recovery methodologies. So we'll cover all type of the required for the cell and gene therapy products. How we are going to culture that. So that also will take up a complete process, and cell processing how we are doing, and cell banking system we are doing. So we'll cover. Already she mentioned as per ICS Q5 guidelines. So we're making master cell bank. So working cell banks. After making these banks, we are going for the characterization. So these and all will cover, and we'll go for the further preparation the purification technologies. So here the manufacturing components and materials will be there. The purified product, how we are going for the formulations. Then I will make a final purified bulk in the formulated conditions, and how will go to treat the patients. So that will be take up preclinical and clinical. So followed by sub-filing into the regulated markets. If we take up, this is a overview, I can explain another 1 hour also, but this is not the time to take up into this level. So the enrolled candidates can take up into, so if you want to looking for the FDA filing, especially for the cell and gene therapy products, so how we are approaching. Before conducting clinical trials, there is a pre-IND meeting. So this will be take up complete requirement for the pre-IND meeting. Then investigational new drug application process and relevant to the preclinical data and CMC data, clinical protocols. There is a day of time required for the review and going for clinical trials to respect to phase 1, phase 2, and phase 3 as mentioned, any risk-based approach, or we can go with any effective to check the conformity and effectiveness. We'll conduct the phase 3 studies. Once this phase 3 studies is complete, how we are going to the biological license application. That is a case study separately. So full CMC data, what is required, non-clinical data, clinical data, labeling information, risk evaluation and mitigation strategies also will be covered here. So like this will take up, so the review process and approvals and standard review will take up 10 months and priority review will take up 6 months. So the advisory committee meetings, facility inspections remain same. How we are doing for the biologics, biosimilars and vaccines, the way of process will be remain same. And FDA guidance on the cell and gene therapy products developers. So FDA provides detailed guidelines. So those are the available. Someone has tested. So where is the guidelines? What are the available guidelines? Yes, those are all available part of our sessions. We can cover it. Yeah, is it clear to everybody?
Yes.
Yeah. So main thing is, so what is the CMC strategies? So that is the main focus. We'll go with that. And respect to the CMC strategies along with the stages of drug development and going for the filing in the various stages. So especially preclinical studies, what is required? What is the relevant CMC data? Then during clinical trials, so what is the applicable CMC data? And filing requirement, what is the CMC data? This will be the majority and filing activities. So clinical and preclinical, we will give minimal and it will be overview, one session like that. Yeah, fine. So we can go ahead. Any questions are there? So yeah, you can share your screen, Dr. Kirtana. Yeah, let's go with the sessions, the questions, anything is there.
Hello, Doc.
Yeah, please.
Yes, I just wanted to find out the differences between how the stem cell therapy works and then the CAR T cells therapy works. And then at a point in time, Doc made mention of the fact that stem cells, as part of the process, undergo seed train, whilst the CAR T cells don't undergo seed train. I wanted to know the differences and why.
Yeah, so yeah, take up, can add up, Kirtana. See, we cannot imagine the way of we are doing for the microbial process and the cell culture process. So this will be a little bit different. So this is an adhesion culture, not in a suspension culture. So that the way of handling will be in the small scales and that will be different process. So if you go with the large scale, that will be challenges. So that's the reason this is in a minimal cases going on, laboratory cases. So like even though in liters, not in the KLs, we are going as of now.
So in the case of stem cell therapy, what happens is you're not taking particularly any kind of cells, right? So they're like you take cells from the bone marrow, the stem cells, where they can differentiate into various cell types. So stem cells are engineered in such a way that you can modify them into different cell types, the type you need. So that is called reprogramming them. So in that case, in the case of T cells, it is very particular, but for stem cells, you can program them into different cell types. So you can use a large scale very easily in the case of stem cells. You just need the raw material in a less amount and then you can proliferate them. So in that case, you need a seed train. Seed train is nothing but you make them in a very large amount. So it's a large scale, you expand them into a large amount. So if in some cases it is adherent, some cases is suspension, then you do it all in a flask. Then you pool them into these bioreactors where all the conditions are maintained, temperature, pH, and all the environmental conditions are maintained. So you expand them in a large scale and then you preserve them, you modify them accordingly, and then you use it for the treatment. But in the case of CAR T cells, you already have very less amount of T cells. So for example, if you take 1 million cells, you can maximum proliferate them, say to around 20-25 million cells max. Then you cannot use them for a very, and then you have to engineer them, select them. So the final product is itself very less. In that case, you cannot use them, you cannot store them for a very long time. So whatever amount of engineered T cells you have acquired, you just reinfuse them into the patient. So in that case, the method is little bit difficult. So but in stem cell, it's more advanced. You can use them in a large scale. So in that case, you need a seed train.
Okay, thank you.
I just want to add one question. Like, how about the scalability of this, treatments? Because if it is like personalized way, how industry is gonna benefit it out of it, and then how it will be scaled to more population? Because as I understand, like it is very personalized and very very small group of people.
Okay, so
It's like as we see biosimilars, we are targeting majority of population, and that way we can make it more higher scale manufacturing, and then that way. So if you see these techniques, they are very personalized, and the scalability, the industrial scalability is I think it's issue. I hope you got my question. Like to make it large scale, these technologies will be difficult. I mean, to cover more population.
okay.
So is it true, or we can scale it at any level?
I think you can scale it to any level because it depends on how many of these patients have these rare diseases, right? So, if it is a very rare disease, then of course you only stick on to that patient. But now most of the genetic diseases are becoming very common, especially in the case of cancer. So blood cancer is getting very more common. Then the scalability is very high. So more of these products are marketed to the patients.
Okay, thank you.
So it depends, I think, on the disease condition, because now researchers are doing it on also solid tumors. So they're trying gene and cell therapy on solid tumors. Till now, everything is on suspended cells. So use on directly into the bloodstream. So you're not doing it on solid tumors. But now researchers are also trying, because that is more in the case of solid tumors are higher population of patients are with solid tumors. So now they are trying, more research is also being done on solid tumors. In that case, then it gets more commercialized.
So, Doc. Hello, Doc.
Yeah.
Yes. So based on the response that you gave with respect to the scalability, coming back to my question about the stem cell and then the CAR T cells, does it mean that if the patients who are having the rare disease is discovered, then that means that the choice of using CAR T cells, because the number is limited, will be preferred. But if the patients who are having the disease are a large population, then in that case, stem cells will be preferable.
Yeah, stem cells is mostly an upcoming, research. They are using it, but if it's advanced stage, mostly it's difficult to treat in any kind. It depends on the level of the disease condition. So where if it is not much advanced, then people can try different methods. But if it is already advanced, then they particularly target it on the cells which has this mutation. So I think it all depends on which stage the disease is.
Okay.
Yeah, any questions from anyone? Mujib Khan, any questions from your end? Yeah, one ready? Pratap, Ravi?
No, thank you. Thank
Are
you,
we the
Kirtan.
session on ready? Are we the session?
Yeah, it's good information.
Hello.
Actually, we are working on mainly biotherapeutics or biosimilars, but this is somewhat,
Correct. So this
of
is,
course,
yeah, this is
basics
what,
we know, but the in terms of therapeutic wise, we have less information on this. So
okay.
it's a good
So
information.
yeah, so you already in batch to participate in the regulatory affairs course. So before
Yeah.
that course and now, how is your balancing of your knowledge?
Yeah, it's good. Then see, of course, I have attended long back, but whatever the things I learned during that session, so now in my current work role, they are very helpful.
Okay.
Understanding the things and then applying some wherever applicable.
Yeah, you people try to attend
Yeah.
whenever possible. I'm
Yeah, sure, sure. But no, because mainly the timing, so most of the times I will reach at 7:38 from to
understood.
home.
By
So
based on that kind point of view, only we are putting
yeah,
session
sure.
at 9 o'clock, not early evening. You know that
Yeah,
everyone is coming from offices, especially for this is for experienced people. Not if in case of freshers means we can connect in the daytime or evening.
yes, yes,
Okay,
yes.
okay,
Yeah.
okay, fine. Thank you.
Okay.
Yeah,
Thank you,
Ravi.
thank
Ravi,
you.
any questions? Yeah, thank you.
Yeah, hi. Am I audible?
Yeah, yeah.
Yeah. Hi, this is Ravi. My only thanks for the in detail explanation of the approach. My question is basically like, how do we ensure the consistency between various batches when we do a scale up or when we are transferring the material? Like, what are the standards we have to consider for the consistency? So, I'm coming from a biosimilar point of view. My question basically, I know that these will have different regulations and different standards, but my basic understanding is like for biosimilar or for any innovative molecule, you'll have certain standards where you'll compare batch to batch variation or lot similarities, anything.
Yeah, see, when coming to the consistency, so whenever we are doing in the GMP activity, so we have to show the validation first thing.
Okay.
So here we are taking a genetical modification cells and we are going for the cell banking and going for the inoculation. Everything will be remain same. So here we are going to take up cell concentration, cell densities. So where you're going to connect in the culture suspensions or maybe adhesion culture and what is the percentage of cells we grown at the end of the stage.
Okay.
And further, once you harvest, then we'll go for the further purifications. And there also we'll see the purification cell separation, especially we'll use the TFF kind of separations.
Okay.
And there we'll see the how the cells are coming recovery point of view.
Yeah, I understand. Like this, my major question is like, what are the quality aspects we have to consider to show the similarity between the lots, like cells being stable, like cells being generated from batches, number. This will give you a quantification. Like,
Yes.
what are the quality aspects? Like
Quality
how?
aspect is one thing is purity and
Okay.
genetic stability.
Okay.
And other relevant to when you cover the purity, will cover all type of viral absences. There are multiple tests, right, regarding adventitious agents. Anything is there to remove that. So what are the controls we have it? So that point of view, it will come like mycoplasma. So other thing point of view like purity by microbial any contamination or that those will be covered here as a quality part, especially for these things. So it will
Okay.
go for further in the molecular level. So when you're going to try genetical mutations, anything happen. So gene copy number, every batch is not possible to test it, but at the initial stage where you're making a banks and RO stage, so that stages will be happen for the validation batches and further will be continued further.
Okay. And in a similar way, do we also check the activity of lot, like lot to lot variation, any activity of the batches?
There are bioassays point of view will happen. So we'll cover in the upcoming sessions because that point of view we quality part point of view will not come. This is an overview, but that is also there. Then only we can come to know the efficacy level.
Yeah,
Because
exactly.
these are the biologics, no? So we had to cover with bioassays to what the binding efficiency is there.
Exactly. Yeah.
So we had to
Thanks
go. So we had to go with the bioassays, so that that we're going to cover in the regular sessions. What are the quality aspects, especially for the cell and gene therapy products?
Understood. Thanks a lot.
Yeah, yeah. Thank you, Ravi, for joining. And Sohal, do you have any questions? Sohal Mujad Khan. Okay, fine. So we will check one by one. Is there any questions? Mujad Khan, no questions. Thank you for Mujad Khan joining and supporting in the LinkedIn. Lot of support from you. I think so. There are messages earlier. I already replied those messages. Yeah, there is no new messages. So we'll stop the recording. Off the recording, any questions want, let us know.